Okay, so today we will be discussing one of the basic topic of molecular biology, that is electron transport system. Now, I hope that before starting ETS you know that ETS only starts when there is a proper concentration of NADH in the cell. Okay. Now you should understand one thing, that it was always thought that how NADH is actually converted into ATP. It was always known that NADH is always converted into ATP. But the problem was there was a gap between how it was converted and when it is converted. That's the very basic thing you need to understand today. Now, ETS is also known by the name of electron transport chain or oxidative phosphorylation. It is also known by the name of oxidative phosphorylation. Oxidative phosphorylation. Now, we should first of all define what is ETS. Now, before that, before defining ETS, we should again understand one thing, that where it is occurring, okay, and then we will define it, and after that we will see that how it is actually operating in the mitochondria. Okay, now listen. The site for ETS is mitochondria inner membrane. It's not actually mitochondria, it's actually mitochondrial inner membrane. Okay, it's actually mitochondrial inner membrane where it is occurring. Now, you know something, that there are two organelles, you know, which has peculiar, you know, structure, and these two are called mitochondria and plastid. You know what is the common between these two? The common thing is that both mitochondria and plastid have double membrane. Okay, these two things, mitochondria and plastid, have two common things, that is double membrane first, and second thing, that is they have their own DNA. That's the two unique things about mitochondria and plastid, you know. Now, first of all, if I want to explain, if I could explain you that why these two have double membrane, then automatically the second part will be explained. Now listen. In the prehistoric times, suppose this is a cell, okay. In the prehistoric times, when the earth was born, actually, you should understand that there was more hydrogen in the atmosphere, okay. As there was more hydrogen in the atmosphere is called reducing in nature, reducing atmosphere, okay. Now, with the evolution of plants, oxygen came into existence on the earth, and in the reducing atmosphere there was many organelle like bacteria. Suppose this is a bacteria, and these bacteria have a special thing, that is they can use these protons from the atmospheric, you know, environment to create energy. They had these already properties. They had already these type of properties, that they can use this reducing atmosphere to create energy. Now listen, you should understand that when Earth went from Earth's atmosphere went from reducing to oxidizing atmosphere, then it became a serious threat for them. Why? Because they are used to this reducing atmosphere and they cannot survive in oxidizing atmosphere, right? Now the problem was solved by themselves, that is, they said to cells that, okay, we will enter your system and we will provide you or we will share with you the energy we were harnessing. Now, when they tried to enter the cell, you should understand one thing. When they tried to enter the cell, they took with them a part of the cell membrane of the parent cell in which they are living. They took with them a part of the cell membrane. They took with them a part of the cell membrane. They took with them a part of the cell membrane Okay. Now you should understand one thing. As they took with them a part of the cell membrane with them, therefore these organelles have two membranes. Okay. Now listen. As they took a part of the cell membrane, therefore they have two membranes. Now understand this very carefully. Before the conversion of reducing atmosphere to oxidizing atmosphere, they were independent organisms. Now you should understand that because they were independent organisms, therefore they had independent DNA. Okay. They had independent DNA with them. Okay. Now, therefore, we can now say that mitochondria and plastid were actually prehistoric organisms, small organisms, which were living freely, but when they actually faced threat with respect to the changing atmosphere, they actually took shelter in our cells and plant cells so that they are saved. Why they can be saved? Because the protoplasm or the cytoplasm of cell have H plus in them. Why? In the biochemistry class we will discuss, because in most of the biochemical pathways, that is glycolysis, Krebs cycle, all these things, the intermediates are actually acids. In most of the cases the intermediates are acids, right? In most of the cases the intermediates are acids, and acid have two forms. Suppose I am telling you phosphoglycerate. Suppose I am telling you what is the difference between phosphoglycerate. What is the difference between phosphoglycerate and phosphoglyceric acid, that is PGA. You know these things. Obviously we will discuss PGA in great details when we will be discussing the dark reaction, obviously. Now you know phosphoglycerate. Now what is the difference between phosphoglycerate and phosphoglyceric acid? The difference is phosphoglyceric acid has intact COOH group with them, but phosphoglycerate has this COO minus, that is, it's an ion form. It has got charge. Remember this for your entire life, because we will need this concept today when we will be discussing something called ATP synthase, which some idiots think that it's an enzyme. No, it's not an actually enzyme. It has some enzymatic properties obviously, but it's not actually an enzyme. Okay. Now phosphoglycerate has negative charge in it, that is, whenever this 8 will be there with a particular organic molecule, that means it's an anion. But phosphoglyceric acid is not an anion. Okay, remember this, we'll need this now. Okay, problem is this: suppose in glycolysis there is phosphoglyceraldehyde, 3-phosphoglycerate, 2-phosphoglycerate, you have written these things in your exams, right? But the problem is this, you never understood that you are writing 2-phosphoglycerate, that means you are already telling that it's in anionic form, the acid is in anionic form, right? The problem is this, phosphoglycerate has come from this, right? Phosphoglycerate has come from phosphoglyceric acid, right? That means how can phosphoglyceric acid be converted into phosphoglycerate? By releasing protons, and these protons are actually taken up by these rascals so that they can use it to harness ATP or chemical energy. Okay, clear? Now there is one theory that is known by the name of, you know, something called I would say Peter Mitchell's theory. Which is also known by the name of chemiosmotic hypothesis, chemiosmotic theory, right? Now remember this for your entire life. ETS has nothing to do with chemiosmotic theory. ETS has nothing to do with that. Listen to me. It has something to do with ATP generation. It has something to do with ATP generation. It has nothing to do with so-called ETS. In fact, ETS has nothing to do with ATP. It is coupled process, obviously. ETS, electron transport system, and ATP synthesis, these two are coupled process, obviously. And that's why we have chemicals like uncouplers, like dinitrophenol, which actually inhibit the process. What is the function of uncoupler? Uncoupler actually uncouples the thing, right? That's why they are uncouplers. That means if you give NADH, you will get ATP. That's the very basic thing, right? But the problem with uncouplers is that you give an NADH, no doubt about this, you will not get ATP because the process has been uncoupled. That means these two processes are not occurring at the same phase. Okay. Now, another myth should be bursted today. That is, Peter Mitchell actually was not the only person for chemiosmotic hypothesis. Obviously, he was the main person behind it, obviously. But it was actually the scientist Boyer who understood his concept and worked on that. It was scientist Boyer who actually understood his concept. He was actually the first person to understand that NADH, you know, oxidation and ATP synthesis are coupled. Obviously, that's true. We'll talk about that in details. But it was not only Peter Mitchell, but also scientist Boyer who understood it and actually studied the thing and actually experimentally proved the thing. And we have very good, you know, wealth of experiments providing the proof of chemiosmotic hypothesis. Now, you should understand again one thing, that what is the structure of mitochondria before we go into the details. Okay, you should understand what is the structure of mitochondria before we go into the details. Now listen. Suppose this whiteboard is a cell and this is the cell membrane. Okay. Now, this is a mitochondria inside the cell and this is the outer wall. Okay. Now, the outer wall of these mitochondria have same type of phospholipid which is comparable to plasma cell membrane. That is, they also are lipid bilayers. Okay. They also are lipid bilayers. Now understand this very carefully, that this first layer of mitochondria is very much unique. Why? You know something called semi-permeability, right? Every lipid bilayer is semi-permeable in nature, not with respect to mitochondria. the mitochondrial first membrane is freely permeable. It is not selectively permeable. That's a very basic difference you should understand. It's freely permeable because they have many proteins associated with that, which actually helps in its permeability. Okay, there are many proteins. There are many protein pumps associated with this. There are many protein channels. Sorry, they are not pumps because pumps required ATP. There are many protein channels associated with this. They carry the work of carrying something from cytosolic matrix, cytosolic area to matrix area. Now, it is freely permeable, but what is the basic difference between this cell membrane and the plasma membrane? Frankly speaking, structurally there are no differences. Functionally there is a difference, that is this is suppose the cell membrane, these are selectively permeable, they are freely permeable. Why they are freely permeable? Not because of their structure, because of these channels. Then what is the basic difference between these two, this plasma membrane and this mitochondrial outer membrane? Structurally there is no difference. That means if you say that there is a structural difference, then probably you haven't understood the words of Robertson. Robertson was a scientist who gave a concept known by the name of unit membrane. There is something called unit membrane concept, you know. There is something called unit membrane concept, right? If you say that there is a basic difference between these two structures, then you have probably not understood the words of Robertson. That is, Robertson said that all the membranes in the world, all the lipid bilayer membranes are of same nature. They have the same nature. Okay? They are structurally similar, but functions will always differ. Why lysosomes, take hydrogen like mitochondria? That's a very basic thing, because they have pumps in it. Why endosomes don't take? Why endoplasmic reticulum don't take that? That's a very basic thing. Why endoplasmic reticulum can secrete calcium? Because they have special pump, specialized protein channels, right? Now, this is the outer membrane. Okay. What about the inner membrane? Inner membrane of mitochondria has infoldings. These are the infoldings. That means it's not a regular, simple. It's not a regular simple membrane, but it has infoldings. Okay, that means the actual size of the prehistoric bacteria was much more larger. That's why these membranes have infoldings, so that they can be fitted in a small space. Okay, now these indentations have different types of proteins which are associated with different types of metabolic process. Or suppose if you want to take in NADH, there is a separate channel for this. If you want to take in citrate, citrate subtle, there is a separate channel for this. That means there are many channels which are associated with different functions. Okay. Now you should understand one thing again, that this mitochondrial inner membrane is actually the place where actually